Otherwise, I just wanted to start today's lecture by saying tomorrow is election day. So please go out, vote, exercise your rights as a citizen. There are shuttles running to and from the voting center from campus here if you are worried about getting to places. Let's see, there's voter information specific to voting as a student on campus at the link that I have posted here. So one thing that I did not know, if that you're a student here, your address is actually your dorm and room number rather than 800 West College Avenue, right? So there's some good information at that link. And then because tomorrow is election day, there is a modified class schedule. I have linked that below. Obviously, it doesn't affect us because we don't have classes tomorrow. But maybe if you have a lab or any of your other classes, you can take a look at that link below. All right. Otherwise, I believe we left off on this slide talking about metaphase. So far we've gone through prophase, we've gone through prometaphase. Right? Prophase, chromosomes have condensed, nuclear envelope is disappearing, and the mitotic spindle has formed. Prometaphase, the nuclear envelope is completely gone. The mitotic spindle is on opposite sides of the cell, and then attached to the chromosomes themselves. That leads us to metaphase. So chromosomes that are attached to that mitotic spindle are now going to line up in a row. And this row we call the metaphase plate, right? So it's not like a tangible physical structure you could identify in a cell. Rather, it just refers to the middle of the cell where our chromosomes line up in a single row. All right. In order to get those chromosomes to line up, remember those microtubules, they are attached to the chromosomes. They're kind of like the puppet strings conducting the chromosomes into place, right? So they're lengthening, they're shortening, they're getting those chromosomes to line up right in the middle. All right, so we've gone through prophase, prometaphase, metaphase. I want us to do just a quick review, all right? So we're going to start basically today with a review after learning about metaphase. I want you to tell me stages of mitosis so far, what has happened?
Where's the room of motor? My brother goes
Down there.
We only got the three of us.
We're running a little behind schedule, so I'm on schedule down the road here. Running a little bit behind.
I think that's okay.
Yes. That was less material for your exam.
Yay.
Exactly.
All right, so stages of mitosis so far. First two we learned on Friday, third one we learned today. What is the first one?
Prophase.
Prophase. What happens to the nuclear envelope?
Dissolve. Starts to start.
Starts to dissolve, starts to disappear. What happens to the chromosomes?
They're condensed.
They're condensing. And what happens with the mitotic spindle?
Begins to form as well.
Begins to form. Yes. So that is all in prophase here. All right. Our next one, what is that called? Prometaphase. What has happened to the nuclear envelope? It's completely gone. What happens to the chromosomes? Well, they're attached to something, right? They're attached to the mitotic spindle. And where is the mitotic spindle located?
The two opposite poles.
On the opposite sides of the cell, yeah, the two poles. Right, so nuclear envelope disappears. Chromosomes have attached to the spindle apparatus, and those mitotic spindles are on opposite sides of the pole. All right, the last one that we just learned about, which is called metaphase. All right, chromosomes have lined up in the center. Right, they're lined up on that metaphase plate. All right, so we still have a couple of stages left. All right, I tried to draw them out on the side, and I will continue my drawing here. So again, you can see nuclear envelope is starting to disappear. Chromosomes have been condensed. You can see them in this X shape. Mitotic spindle has formed. In prometaphase, mitotic spindles on either side of the poles, or either side of the cell at the poles, and they are completely attached to either side of the chromosome. Then in metaphase, those chromosomes are all lined up in a single row on the metaphase plate. All right, so let's move on to anaphase. All right, so in anaphase, we're going to split apart our chromosomes. Right? So at this point, we have the mitotic spindle. It's on either side of the chromosome. It's attached to either side of the chromosome. You're now going to split that chromosome in half. Right, so we're splitting the chromosome and we're separating out those sister chromatids, which is either side of the chromosome. So if I were to draw out anaphase, and my mitotic spindle is still at the poles, the length of the microtubules have shortened a little bit because they have pulled the chromosomes apart. And then the chromosomes themselves are no longer in that X shape. They're single chromatids. We have split the sister chromatids in half. All right, so we haven't split homologous chromosomes. We still have homologous chromosomes, right? They're just not replicated anymore. Right, so greens are homologous, oranges are homologous in my diagram up here. In the diagram up top there, they're not color-coded, but rather based on size, right? It's harder to tell in that diagram. But what we have split apart are those sister chromatids, right? So again, those puppet strings attach to the chromosomes. They have started to shorten, and that breaks apart our chromosomes into two pieces. All right, now as they're breaking apart, right, they're moving to opposite poles, right? They're being pulled to either side of the cell. In our last stage, they have reached the opposite side of the cell, right? So our last stage is telophase here. Our chromosomes, which have already been split apart, are reaching opposite sides. Right. They are detached from the mitotic spindle, right? We don't need to move them around anymore. They're already to where they need to be. Because they're at opposite sides, that nuclear envelope is going to start to reform, but now we're going to form two nuclei, right? One for either cluster of chromosomes that have been pulled apart, right? So it's going to start to reform at the beginning of telophase. By the end of telophase, those two nuclei are fully formed. So essentially what we're left with is one giant cell with two nuclei, where that genetic information is evenly divided between those two nuclei. All right, does anybody remember what happens next? Splits, what do we call that? Cytokinesis. So the last thing that has to happen is cytokinesis. Now cytokinesis is going to start during telophase. And we can see that in this image up here. Right? We can see this cell is already looking like it's starting to split into two. So cytokinesis has already started. Right? Now, cytokinesis, whether we're talking about plant or animal cells, is going to occur no matter what, but they're going to occur in slightly different ways. Right? So in an animal cell, it essentially creates like this pinch point. We call it the cleavage furrow because we're cleaving two halves of the cell. Right? So we'd have basically this pinch point here, right? And then that pinch point will keep pinching until we get two completely new cells. Now a plant cell, if you remember the exterior of a plant cell, they have a cell wall, which animal cells do not. And that cell wall is rigid. So rather than trying to pinch apart, right, this really rigid cell wall, it's going to go through cytokinesis slightly different in that it produces a cell plate, right? So that's in the middle of those two nuclei. This plate begins to form, right? And then when it's fully formed, it creates a new plant cell wall, creates two new cells. So same process, right? Splitting apart the cytoplasm, creating two new cells, but in slightly different ways. All right, so we have now learned all the stages of mitosis. Remember mitosis, we're going from a diploid cell to a diploid cell. They are genetically identical to one another. So before we complicate matters by bringing in meiosis, I want to make sure we fully understand this. So we're going to do a review. And I'm actually going to have you do a review of 2 slides, and I'm going to give you about 10 minutes. So first of all, I want you to tell me what major events are happening in each of these steps. Then I want you to look at this image and try to figure out, right, based on this light microscope picture here, what these cells are going through, what stage of mitosis, or if it's going through interphase. Right? And then finally, try to come up with a mnemonic to remember the stages, right? Remember the order of those steps. And again, I'm going to give you like 10 minutes for this because I want to make sure we understand this before we move forward. So if you don't have protein, those are dense, but they're yeah. Still trying to scramble together on these. Yeah, you got it. Yeah. Call, so you could also say, like, because there wasn't, I was like, yeah. Right. You're doing better than my other section.
I mean, it's kind of straightforward. Just think about it right.
Yes. Yeah, I think that's a good way to put it.
Because it looks like this actually makes sense.
Yeah.
You know about that? Alligator god, Zeus that they got him the big fast boat. There's this thing like fast pro shops. Oh yes. It's in. My dad stays at home or like just on like
Let's just hope it's not
I want that's two has to be sent. Modern wonder of the world. One more fan.
I like the
Yeah. Release. I said five. Yes.
Five is, I think, the tricky one.
Like the description. I'm saying like this slides, or like you know how we have like
See, this stuff is like the only stuff that actually makes sense to me.
Good.
All the other stuff is.
Yeah.
It's like, okay.
'Cause I think in every biology class, you can't get through a class without talking about mitosis at the very least.
I also just didn't have a really good science program in school.
Yeah. Yeah, that happens. I also grew up in a very rural, small town. We didn't have many options.
I try and like we not
Gotta keep going.
So those five.
Discussion will come here. Yes,
We so we did like sort of
yeah. I got 6 packs. Your knowledge.
so we say those five. We go to those five.
They don't get to understand what happens. Like, you know what, I can't do that to them. Or not on purpose. I would say my thing. Yeah, so it's gonna be funny because my entire I could if I didn't have to grab it. I know there are people who have like dragons.
So what would you, like prophase, how would you?
It's, I mean, most of them can't have two. I'm only allowed to have 2 rabbits because I had a saying that I can't say. No, my friend. One day got off of work.
All right, let's come back together here. So we kind of already did this review for the first 3, right? We looked at prophase, prometaphase, and metaphase. So prophase, we know that the nuclear envelope is disappearing. So the chromosomes, there you go. Chromosomes have condensed, right? And the mitotic spindle has formed. Then we went through prometaphase and we reviewed that as well, and we know that the nuclear envelope is completely gone at this point. The mitotic spindle is attached to the chromosomes, and they're at opposite sides of the cell, right? So they're at the poles. I need to charge my pointer. Can't stand too far away. There we go. And then metaphase is the last one we went through, right? The chromosomes, they are lining up on the metaphase plate. What is happening in anaphase? What's happening to the chromosomes?
The connection between them are being broken.
Yeah, they're being broken. What is being broken? Like, what is separating apart?
Sister chromosomes.
Yeah, sister chromatids, right? There's a very specific word for that. That's a great question that is likely to be asked on an exam, right? At which phase in mitosis, right, do the sister chromatids break apart? And maybe how is that different from meiosis, which we will learn a little bit about later on. All right, anaphase, they have split apart, right, and they're moving towards the poles. And then finally we have that telophase and cytokinesis. Right, the big thing in telophase is that the nuclear envelope has reformed, right? The chromosomes are starting to decondensed. Okay? And cytokinesis is splitting it into two. All right. After class, I'm going to make these answers available onto your slides because I know it's a lot of text, right? But we're going to move on to those images because I really like this question. It puts your knowledge into a practical application. Like we could be looking at a microscope at actual cells and figuring out what they're doing in terms of mitosis. If I were to look at number 1, what phase might you say number 1 is? I would say it's prophase. Yeah. What clues led you to that? It's messy. Yeah, it's messy, but you can start to see individual chromosomes, right? Chromosomes are condensed, much more condensed than maybe some of the other cells around it. What about number 2?
Telophase and cytokinesis.
Yeah, telophase and cytokinesis. So you should have picked up that here we have 2 different nuclei. Now the view that we're getting of those nuclei is like if you were looking straight on, this was a nucleus and this was a nucleus, they're kind of overlapping, right? But they are 2 separate nuclei, right? But it looks like it's still in one cell, so that would be telekinesis and, telophase and cytokinesis. There we go. What about number 3?
Metaphase.
Yeah. Metaphase, they're lined up in a row, right? Number 4, anaphase, because we can see they're still kind of lined up in this orderly fashion, but they're being pulled to opposite sides, right? Number 5?
Interphase.
Number 5 is interphase. Yeah. So if we look at it, we see our chromosomes here, right? But they're not really condensed. You can't identify a singular chromosome apart from another one, like maybe you can in prophase. In fact, many of the cells in this image that I did not circle are in interphase. And that leads us to 6. What do we say number 6 is? Yeah. Prometaphase or early metaphase, right? They're condensed. They're definitely connected to a mitotic spindle at this point, even though we can't physically see that spindle. But we see they're starting to become organized. Remember mitosis? Right? There's no clear ending or start to each of these phases. They each flow into one another. Did anybody come up with a mnemonic to remember the order in which these phases go? No? Alex, I think you heard the one I was going to say. A PP math, right? It's silly, it's stupid, it's like you're potty training a dog, right? But it's a PP math. Prophase, prometaphase, metaphase, anaphase, telophase, right? So that's how I remember it, right? There's many other mnemonic devices out there that you might want to use. Any questions before we continue? All right, so we've gone through mitosis, right? We know mitosis can occur via asexual reproduction. So if an organism asexually reproduces, all right, it's not mating with another organism, it's just creating a genetic identical copy to itself. Right? So mitosis is the way that asexual reproduction is done. Right? I have an image up here of that hydra. Once again, I know I mentioned the hydra last week as well, but they're really good examples of creating new individuals that will eventually break apart and become their own organisms themselves, but they're genetically identical. Versus sexual reproduction, right? This involves the formation of gametes that will fuse together to form a zygote. Right? And the key here is that the offspring, not genetically identical, and in fact, there's a very unique combination of genes. We're getting genetic diversity. So sexual reproduction, these organisms are going through meiosis. Asexual reproduction is done via mitosis. I'm going to pause just a little bit because I know there's a lot of text here. I could have also put, instead of this hydra, if anyone's into like houseplants and propagating new plants, where you pluck a leaf off of a plant, stick it in dirt, and that leaf creates another individual. That individual is genetically identical. That's another example of asexual reproduction. All right, so mitosis, asexual reproduction, meiosis, sexual reproduction, because meiosis leads to gametes. Now, why bother, right, with sexual reproduction if you can just asexually reproduce, right? Well, there's disadvantages and advantages to both. Some of the disadvantages of sexual reproducing in the animal kingdom: it's complicated, it's costly, right? You have to invest time and energy to find a mate. I don't know if you've seen some of those mating videos of birds in like the tropics, where they gather a bunch of like items that are blue and then have elaborate dances to find a mate, right? So there's a lot of investment into mating strategies, right? It's also potentially dangerous, right? So when you're putting yourself out there to find a mate, right, you're visible to predation. Think about all those mating strategies. I always turn to the birds because the birds just have these elaborate dances in the tropics, right? But if they're putting on this whole production, this whole show, they're very visible to predation, right? And when they mate, right, they don't know what they're getting themselves into. They might be getting a disease from their mate, right? There are potentially dangerous mechanisms at play. And then it's not always successful, right? Sometimes, right, in the animal kingdom, you mate to produce offspring to preserve your genes, but it's not always going to produce offspring. It's not always successful. Lots of disadvantages, but there's one advantage, and this advantage is huge, right? We are increasing genetic variation, right? We're increasing diversity between individuals and within populations. So an example I like to use for this one is bananas, right? So the banana plant they're all basically genetically identical to each other. The bananas you buy in a store are clones of other banana plants that you would buy in a different store, right? They're all genetically the same. The problem with that is that if a disease comes along, and there are some diseases specific to bananas, it could wipe out the entire population of bananas because there's no genetic diversity, right? If one is susceptible, they're all susceptible. Versus if there's genetic variation, right, maybe some would die, right, but others would have genes to resist those diseases. So having this genetic variation, this diversity, is very critical to the survivorship of offsprings and populations. All right, so in sexual reproduction, we are often going to alternate between being a haploid cell and a diploid cell. And for some individuals, you might spend most of your time as an organism made up of diploid cells, or an organism made up of haploid cells, or maybe you'll alternate between the two stages. So there's these different types of life cycles that I'm just going to touch upon, because if you take organismal biology next semester, you will dive even deeper into this here. But we could say we could be a diploid dominant species. So most of the time, all of our cells are diploid. It could be a haploid dominant species. Again, most of the time, our cells are haploid. Or you can alternate between the two. So if we look at examples of this, right, most animal species, us included, are diploid dominant species. Right? You take a selection of cells from your body, they are going to be diploid cells, right? Most all of the time. The only time our cells are going to be haploid are in our gametes. Right? So most of the time, we're always diploid dominant in our cells. Other species, like fungal species, when we see fungi outside, right, growing in the bread you might have left out too long, right, most of the time they're going to be haploid dominant species. So if you collect the cells from the bread mold, you're likely only going to see haploid cells because they're a haploid dominant organism. Plants have a unique strategy. They will alternate between being haploid and diploid at different parts of their life. Right? So they can go from a diploid plant, go through meiosis to create haploid cells. Those haploid cells will go through mitosis to create a haploid plant. Eventually, those haploid plants will produce gametes, they'll fertilize and create another diploid plant. Right? And the cycle continues, they alternate between the two. All right, so lots of different life cycles out there, life strategies for other organisms. All right, but the key here is at some point there will be haploid cells, and at some point there will be diploid cells. So if we're talking about meiosis, we're talking about the production of haploid cells, or gametes. All right, so meiosis, we are producing haploid cells from an original diploid parent. Okay? And those haploid cells are gametes, right? So in mammals and humans, that would be egg and sperm. In other organisms like plants and molds, that might be pollen and spores, right? Anything that's haploid and is going to eventually fuse with another haploid cell would be a gamete. Right? During sexual reproduction, two of these gametes, they will come together and fuse into one cell that we would call a zygote. And from the zygote, we would go through mitosis to develop a whole new individual. Right? So in humans, right, egg and sperm, we have 23 pairs of chromosomes. In our gametes, we just have 23 chromosomes. We don't have pairs because they're haploid. So a haploid cell meets a haploid cell, right? When they fuse together, they create one cell that is a zygote. That zygote is now diploid, right, because it has a matching pair. And we say it has 46 total chromosomes, but they're in 23 pairs. Right? So after a zygote, then again, mitosis will happen. The organism will grow and reach maturity. Right? But we have to produce these gametes. Right? And these gametes are going to be produced via meiosis. Right? But before we move on to that, I just want us to do a quick review. Right? What are the advantages of asexual reproduction? What are the advantages of sexual reproduction?
Wait, what if humans were asexual? There would be no genetic diversity, and if
I don't care. Explain to me. It's not like
Good question. Good thing to think about.
it's trying to pronounce. I was explaining to them.
because if they were asexual, how could they be more genetic diverse? You get the right to hold why we can populate fast. Just like no, it can't because we are asexual. I feel so good right now.
I have a question. So, if kind of like the advantage of sexual reproduction is like, obviously probably like what? How are bacteria able to like the work on and stuff like that?
Well, guess what our biggest
No pile. That is for me. They already agreed. They already agreed.
you see,
You already agreed. You did it, but everybody else did.
everybody knows it's weird.
For me, it's no.
That's not. You're in our league.
That's fine. Yeah, we're also halfway through the point.
I started one guy out. Just got out right before the game.
Most seed accumulation
7 and 2. Patrick was 7. I think I still have it.
I think I'm 4.
So like, since reproduction we have more degree,
I just
or is it kind of like more free volume?
Yeah, their lab
millions per hour.
When did you
Like spread some cells about. All right, so what are some of those advantages of asexual reproduction? What's the advantage of not needing to find a mate? Is that what you were going to say?
Yeah.
Yeah.
Sorry.
You don't need to find a mate. Why is that? Why can that be a good thing when producing offspring?
Finding this hard.
It can be hard. It's costly. It's a lot of time investment. It's a lot of energy investment. Yeah. So basically, if we go back to that slide where I listed the disadvantages of sexual reproduction, those would all be the advantages of asexual reproduction, is that you don't have to do that. What about the advantage of sexual reproduction? And this is a big one. What was that?
Genetic variation.
Genetic variation, yes, it's huge. We're getting genetic variation. We're increasing the diversity in our population, right? And diversity in a population makes that population more resistant to change, right? We're not just going to wipe out the human race in one go because there's so much genetic variation. All right, so let's talk about sexual reproduction and creating those gametes, right? So that would be the process of meiosis. So on this slide, I just have a broad overview of meiosis. So again, we're going to start with interphase. Interphase is going to happen. We're going to duplicate our genetic information. But then meiosis is split into two. You would say meiosis 1 and meiosis 2. Meiosis 1, we're going from a diploid cell to a haploid cell. Right? We're separating homologous chromosomes apart, and we're exchanging genetic information. The result of meiosis 1 would be cells that still, even though the homologous chromosomes have separated, we still have sister chromatids. It's still in that X shape. Meiosis 2, we're taking the resulting cells from meiosis 1 and basically going through those same steps to create 4 daughter cells. Each daughter cell, right, has 1 sister chromatid in it. So we're separating out the sister chromatids. So in meiosis 1, we're going from diploid to haploid, separating out homologous chromosomes, homologous pairs, and exchanging genetic information. Then we're going to go straight into meiosis 2. Right? We don't have to go through intraphase again because we don't have to duplicate our DNA. Straight into meiosis 2, those haploid cells are going to create more haploid cells, but now the resulting cells are going to only have 1 sister chromatid in it. We're splitting those sister chromatids. All right, we're going to go in depth, at least in meiosis 1, because it's very different from mitosis in a lot of aspects. Meiosis 2 tends to be really similar to mitosis. I'm going to give it a nice long pause here. I see a lot of pencils furiously writing. As we go through meiosis, pay attention to when the ploidy level changes, when we're splitting homologous pairs, when we're splitting sister chromatids. These would all be the types of questions I might ask on an exam, right? In what phase do we split homologous pairs in meiosis? Or how is meiosis different from mitosis? Or like some big changes? If we start at the first step of meiosis, we're going to start at prophase 1. All right, so notice meiosis 1, prophase 1. A couple of similar things are happening here. All right, chromosomes are condensing, just like we saw in mitosis. The nuclear envelope is starting to break down, just like we saw in mitosis. What's different is that now we're getting a structure called a bivalent forming. Right? So bivalence are homologous pairs that are joined together. I'm going to represent a homologous pair by length here, and color represents which parent it's from, mom or dad. When we form a tetrad, we're essentially pairing pairs together, like we're physically attaching them together. So if I were to draw it out, I would draw something like where they're right up next to each other, right? Because they're physically attached. We would call this a bivalent. Sometimes it's called a tetrad, just depending on the textbook and terminology that you're looking at, right? And they form into this bivalent in a process called synapsis. Right? Synapsis is when they're physically joined together. Now, the formation of these bivalents are pretty important because they lead to exchanging of genetic information. One of the main ways that meiosis is different from mitosis. All right, so when they exchange genetic information, we would call this process crossing over, where segments of chromosome are swapped with other chromosomes, all within a homologous pair still. And we're going to go over this step by step because I want to make sure we understand this. All right, prophase 1, lots of similarities to mitosis. Biggest difference, we're forming tetrads. Crossing over is occurring. Let's talk about crossing over. Right. Another word for crossing over would be homologous recombination, right? Because we're taking homologous pairs and basically cutting them apart and recombining them together. And if we know homologous pairs, right, we know the definition is that they contain the same type of genetic information, but maybe slightly different variations. So those different variations we would call alleles. So if we look at this image here, these are two homologous chromosomes. They have not been replicated yet because they are not in that X shape. At certain points on these chromosomes is where we would find the DNA code for different genes. This gene here is about earlobe attachment, right? Whether your earlobes are directly attached to your face or whether they hang down. The different types here, right? The allele for attached earlobes or the allele for free earlobes, these are different variations on the same gene. Right? So we would call these our alleles. A lot of very interesting alleles that we can physically observe on each other. One of them is earlobes. There's another one about hairline. One's like a widow's peak, one's not. Whether or not you have a hitchhiker's thumb. I don't, but if you do, your thumb can hyperextend backwards. Right, those are all different variations of a gene. All right. So if we look at homologous combination, we're basically swapping around our alleles between pairs. Okay? Homologous recombination only happens between homologous chromosomes, not between sister chromatids. Okay? So remember, a sister chromatid is either side of that X. Right? If I were to break apart the chromosome, swap it around, it would not make a difference because they're identical to one another. Rather, we want to swap around information from a homologous pair. Right? So if I have my homologous pair here and I decide to swap segments, right, I might get a result that looks something like that, right? Where we're swapping the alleles around, right? So this produces what we call recombinant chromosomes. The result of crossing over are these recombinant chromosomes, right? We've changed the collection of alleles that each chromosome has. So what recombination does is it increases genetic variation. Right? Instead of guaranteeing that each sister chromatid, identical as they are going to split and go to different cells, right? Now we're changing around some of that information on the sister chromatids to increase genetic diversity even more. So what it does not do, it does not create new genes, and does not increase or decrease a likelihood of a mutation. But what it does do is create new combinations of genes, right? Again, if we look at my example up on the board here, instead of this purple chromosome always giving away a solid purple half to the offspring, now it's giving away, maybe one will be a solid purple half, but the other one would be purple and red. New combinations of those alleles. All right, so lots of variation being produced here. I want us to walk through recombination step by step with a little bit more detail so we understand what's happening. All right, our homologous chromosomes. In our image here, they are denoted as being blue and red. Right? Maybe blues for mom, reds for dad. They are in what's called a tetrad. So homologous pairs are joined together in a tetrad. And on each chromosome, we have different versions of alleles. So at the top of this image here, we have a red chromosome and a blue chromosome. On the red chromosome, we have the capital allele, so capital C, capital E. On the blue chromosome, we have the lowercase alleles, so lowercase c, lowercase e. Right? Maybe C refers to hair color, E refers to eye color. When they're in the tetrad, they're going to break segments of their chromosomes apart. You can see that in the second image here. Inside parts of this chromosome, on the longer half, they are being physically broken apart. But as soon as they're broken apart, they're joined back together. They're kind of twisted back together in what's called a chiasma. Right? So this chiasma is being formed. They're twisted back together. And they do a flip-flop. Right? So now the blue switches sides to the red chromosome. The red segment switches sides to the blue chromosome. And what's left are these recombinant chromosomes that now have different variations, different combinations of our alleles. So now instead of red being all capitals, right, it has a lowercase e. And instead of blue being all lowercase, it has an uppercase e. Right? New combinations have been created. This will go on to split apart even more in meiosis 2. When at the end of meiosis, the result will be that each sister chromatid up here is in a different cell, right, creating different combinations. So instead of red always being capital C, capital E, now we have one that's capital C, lowercase e. Right? New combinations have been created. All right, are you all still with me here? Sort of. We used to have a lab that we would teach concurrent with this section that reinforces these ideas, but we don't anymore. So I'm really curious to see how we're going to do this without that lab helping us out. So if we need to pause and slow down and go over this with a hands-on activity, we will. All right, let's do a review. I want you to answer the questions. All right, what stage does meiosis crossing over occur? Does it happen between sister chromatids or homologous pairs? And then what is the purpose? Following along?
Yeah.
I'm one of those learners where I can't take it in the learn it and then go back. Maybe. You never know.
We'll probably go back and read it out loud 5 times.
Watch the videos like this and they help you.
I actually go through I have back. I don't take it to I do. I just have right now stuff and then I'll add to it.
All right, if I were to ask you on an exam, in which stage of meiosis does crossing over occur, what would you tell me? Prophase 1, yes. If you said prophase, you might get partial credit, but you want to get full credit until you said prophase 1. All right, does recombination happen between sister chromatids or homologous pairs? Homologous pairs, yes, because there is no point in swapping identical information between sister chromatids. And what is the purpose of recombination? Increase that genetic variation. Yes, that's one of a couple of ways that genetic variation is increased in meiosis. All right, so that was still all prophase 1. Prophase 1 has a lot going on. Now if we move into these second steps, we're in prometaphase 1. So again, there's some similarities to mitosis. Right? At this point, the nuclear envelope has completely left. It's gone. It's no longer there. Spindle apparatus is complete. Right? And the spindle fibers have connected to our chromosomes. This is where the similarities stop. Right? The spindle fibers are now connecting to opposite sides of tetrads rather than opposite sides of a single chromosome. So if we look at our image up here, we have our bivalents, right? Crossing over has occurred. The bivalents are still together, and the spindle fibers are attaching to either side. Versus if we remember mitosis, right? This bottom one is now showing mitosis again. We would have our chromosomes not in bivalence, and a mitotic spindle would be attached to either side of all the chromosomes. Right, so that is a key difference there, attached to bivalence versus individual chromosomes. All right, so if I were to draw it out, I drew too many down here. Right. My mitotic spindle would be attaching to each side of the bivalent. Right? And in this case, I would have one bivalent, so only one mitotic spindle. Versus in mitosis, we saw it connecting to either side of both chromosomes. All right, so key difference here. So now our sister chromatids are attached to a single pole. Right? One side of the bivalent is attached to the spindle mitotic fibers of the other, of one pole, and the other sister chromatids are attached to the spindle fiber of the other pole. All right, so now they're attached. We gotta line them up. And we're gonna line them up in metaphase 1 again, okay? But they're gonna line up in a different way. Because they're tetrads, they're still tetrads. So in mitosis, we have them lining up in a single row. Right? So if we thought about mitosis, our chromosomes would be lined up in this single row. But now we have tetrads, So we're going to line up our chromosomes as tetrads on the metaphase plate. So it almost looks like it's a double row of chromosomes. Right, so that is another big difference between mitosis and meiosis 1. Right? So now we have a ring of tetrads on the metaphase plate. Now this is going to lead to another way that genetic variation is increased in meiosis. But we're going to talk about that on Wednesday because this is also where my last class left off. So I'm going to stop us at the same point here. So we will stop at metaphase 1 and then continue with this idea of independent assortment on Wednesday.